Hey, we are going to study about the DNA and its packaging. What is a DNA? This is the molecule you must have seen in your biology textbooks. This is what a DNA looks like. But what does it do? Suppose there are two guys fighting that this is their son. He says, he is my son. He says, no, he is my son. Now to test, we can do something called DNA testing. And on testing, if I find that this guy's DNA matches with this one, so we say he is the real father of him. Because we believe that during these sexual cycles, we give our DNA or the genetic material to our offsprings. So that means this guy must have given its DNA to his son. And along with the DNA, we also give the characters, the hair, the skin color, the eyes. That is why we look somewhat similar to our parents. Hence we can say that DNA is responsible for the inheritance and is the genetic material for us. Now, where is DNA present and how is it packed? DNA is of course present inside the cell. What are the types of cell we know? We know prokaryotes, we know eukaryotes. Now at this moment, I want you to pause the video and think the major difference between prokaryotes and eukaryotes. Have you thought? Okay. Now what does a prokaryote have? And what is the difference that makes prokaryote and eukaryote different? So to draw a prokaryote, we draw a cell wall, we draw a cell membrane, but do we draw a nucleus? That is what it makes difference. Prokaryote does not have a nucleus and they have a nucleus. This is a very big difference between them. Okay. So now, where I will draw the DNA here? I will draw the DNA scattered like this. But why didn't I draw on this DNA scattered all around the cytoplasm? Why I did not draw it scattered all around? Can you tell? This is because it is attached at this place by some beads. I am saying it bead, but it's actually a molecule. We call it protein. And these proteins attach the DNA with them. Hence DNA is not scattered throughout it. What is the charges on proteins? These proteins are positively charged. What is the charge on DNA? DNA is basically negatively charged. How can you remember this? You can remember this by P for positive, N for negative. What do we call this thing? Because it is not a nucleus. It is of course not a nucleus because there is no membrane. What will I call it? It's called nucleoid. Not a nucleus, but a nucleoid. So it's very easy packaging in prokaryotes. But if we talk about the eukaryotes, the complex ones, they have a very complex mechanism of packaging their DNAs. So this is the cell membrane and I am drawing a nucleus here. Because the DNA in the eukaryotes is present inside the nucleus. So I am drawing the thread of DNA. Suppose this is the DNA threads. They are very highly condensed. These condensed threads are called chromatins. And during this cell division, we don't see these condensed threads. During this cell division, we see that these threads are arranged in some structures like this. They are called chromosomes. Now, I am showing you the zoom in part of the chromatin. So let's take this thread out and draw it here. When we look it closely, we see that this thread have the presence of some beads. Beads in a string-like appearance. And if I zoom this bead, I see that this bead is made up of 8 molecules. 4, and I am drawing 4 molecules behind. Okay? So it is made up of 8 molecules. Now, if there is an 8 molecular structure, we call it an octamer. But what is this thing? Is this a protein or a DNA, or what is this? This green color thing is actually the protein, as you have seen in prokaryotes. But this protein has a name. This protein has a name called histone protein. And this thread is the DNA. What is the charge that these things have? I told you P for positive, histone protein is positive, and DNA is N for negative. Negative attaches to positive. Same thing applies on prokaryotic packaging. Now, if I ask you, what is histone protein's nature? Is it acidic or basic? So proteins are basically made up of amino acids. If I check the amino acids nature, I can tell what is the nature of histone. So these amino acids which are present in histone are lysine and arginine. And lysine and arginine are basic amino acids. If they are basic amino acids, it means the histone protein is basic in nature. So what will be the nature of DNA? You can get it from its name itself. Deoxyribonucleic acid. Hence it is acidic in nature. Okay, so we have come so far through. But do you think there is some mistake? I have drawn the DNA double stranded in that figure. Here I am seeing it one stranded. Is it a wrong figure? Pause the video and think about it. No, it is not wrong because I have not zoomed in completely. Now I am zooming in further. So I can see, yeah, there are two strands. Okay, I have joined these two strands. Actually, when I open this thread, we see that yes, there are two strands. DNA is actually a double-stranded molecule. A part of DNA is called gene. So this is a part of DNA called gene. Here are some things to keep in mind. That histone protein on which this DNA is wrapped around is an octamer because it is 8 molecular structure. And also, if we talk this unit of the one thread and this histone protein of histone octamer wrapped around by the DNA, this molecule is called nucleosome, this unit actually. Okay, now let's discuss the packaging at higher level. I'm going to draw this thing. This is a structure we see during the cell divisions. Okay, I'm drawing it a more grader picture. Okay, so it contains some proteins which are not made up of histones. So this protein is not made up of histone. Okay, so this is called NHC, non-histone chromosomal protein. Okay, non-histone chromosomal protein. Now I am going to wrap this molecule by this chromatin. I am closing it DNA again. I am zooming it out and I am wrapping it, and now it is wrapped on it like this. This is the highest level of packaging present in the eukaryotes. This whole chromatin thread is wrapped on it. Okay. Oh, I am missing something, yeah? Am I missing something? Yes, I am missing the histone protein. See, I didn't mention these beads. Yeah, so there are beads which I have not mentioned on zooming it out. So histones and this chromatin is all wrapped around NHCs. Now this packaging may be loose or dense. If the packaging is loose and on staining we see a light stain, then we call the chromatin as euchromatin. If the packaging is very dense, then on staining we get a dark stain, and such a chromatin is called heterochromatin. This was the most highest packaging and all about the DNA packaging.